Welcome to our video lecture on topic 2.7 DNA replication. Our essential understanding for today: genetic information in DNA can be accurately copied. Today we're looking at how do we take DNA and copy it into more DNA. Our objectives: we're going to review DNA structure super briefly. We're going to describe the Meselson and Stahl investigation that helped us understand that DNA replication is semi-conservative. We're going to state the functions of the enzymes involved in replication, and then we are going to wrap up with a little explanation of PCR, polymerase chain reaction. This little bit of a review: structure of DNA. Remember that DNA is a double helix. So I have these two strands. Those strands have these backbones of sugars and phosphates, sugar-phosphate backbone. And then in the middle, we have those nitrogenous bases. The strands are anti-parallel. That means they run in opposite directions. So here, I'm going to call this the 3' end of this strand of DNA. The reason that it's 3' is because we do love to count our carbons in biochemistry and organic chemistry. Carbon number 1 in the deoxyribose sugar molecule is bound to that nitrogenous base. Carbon number 2 is here, 3 is here, 4 and 5. 5, remember, is always next to the phosphate group. So this carbon number 3 is the end of this strand of DNA, and so I'm going to call it the 3' end. At the opposite side of this strand, we actually end with carbon number 1, 2, 3, 4, 5. 5 stuck to the phosphate group, so here's our 5' end. My other strand, the opposite side, runs in the opposite direction. So where I have my 5' end here, I have my 3' end here. Here's carbon number 1, 2, 3, 4. That one's 5 next to the phosphate group. And then, of course, at the opposite end, I've got carbon number 1, 2, 3, 4. There's carbon number 5, and there's my phosphate at the end of the 5' end. These two strands are held together by hydrogen bonds between complementary bases. Remember that adenine and thymine are complementary, and so we have some hydrogen bonds that hold them together, hold those double helices into double helices instead of just singles. And then our other complementary pair in DNA is guanine and cytosine. So I've got my guanine here, cytosine here, and they are also held together by hydrogen bonds. DNA replication is what we call semi-conservative. That means that half of the old DNA, that's the semi part, is saved, that's the conservative part, in the new DNA. When I look at this lovely model of DNA, this blue DNA strand is our original DNA. It's our template DNA. Each new double helix, notice, is half blue, and then half of that new red DNA that has just been composed. This guy also half blue and half red. So we're going to save, that's the conservative part, half of the original DNA into each new double helix. You might be wondering, how do we know this? These guys, Meselson and Stahl, figured it out. So they were looking at 3 different models for DNA replication. The first one was conservative. Conservative, like literally we're going to save the whole piece of original DNA. So this one became all the same. This one stayed all the same. We made a new version that was completely new. Semi-conservative, what we just looked at on the previous slide, just half of it is saved. Here's our original DNA. When I go through that first replication, I have one double helix that's half of the old, and one strand of that double helix is half new. Same thing for the other guy. Dispersive, this one's kind of crazy. What if this original piece of DNA just popped apart into a bunch of pieces, lots of fragments, and those fragments got mashed together with fragments of new DNA like a jigsaw puzzle? It's an interesting theory, but kind of crazy, right? How they figured out that semi-conservative was the winner is they grew bacteria. Bacteria has DNA. They grew that bacteria with different isotopes of nitrogen. We have some nitrogen called nitrogen 15. Remember that isotopes have different numbers of neutrons and therefore different masses. This guy has 7 protons and 8 neutrons, which is where that 15 comes from. We have other isotopes of nitrogen that are nitrogen 14, 7 protons and 7 neutrons. That makes this guy have a total mass of 14. All of the blue DNA was grown with nitrogen 15. And so the bacterial DNA had this heavier nitrogen isotope. We're able to separate the different nitrogens, the different DNAs with the different nitrogens, by sinking them in centrifuges. The heavier nitrogen DNA sank a little bit more in the test tubes than did the lighter nitrogen. And so what Muselson and Stahl did was they let the bacteria grow. They let it make some babies, and so we had some DNA replication happening, and they sent this DNA through test tubes, and they noticed that it was kind of a mashup of 14 and 15. And then they let the bacteria grow a little bit more, and they noticed that again, there were some mashed up pieces of nitrogen 14 and nitrogen 15. But in that second generation, there were also pieces of DNA that were only nitrogen 14. And that led them to the understanding that DNA replication is semi-conservative. We're going to talk about replication, all the details, all the enzymes involved in replication now. These are some of the enzymes involved in replication. These are the ones that we're going to talk about. There are more. And these are mostly bacterial enzymes. There are different ones in eukaryotes, but this is where we're going to start. So this is a good start for us to learn lots about replication. First up, we're going to look at this enzyme helicase. Helicase's job is to unwind the double helix. We're going to take double and turn it into single, and that happens by breaking hydrogen bonds between those complementary base pairs. Here's our helicase. We have some complementary bases here. Helicase's job is to come in and break those double helices, break the hydrogen bonds, holding the double helix together so that we have some open space so that we can actually make copies of it. It's hard to make copies when things are all wrapped up super tight. Once helicase breaks those hydrogen bonds, it's super easy for them to reform. So we add these guys called SSBs, or single strand binding proteins, and this just helps to keep the double helix single helix. We don't want it to zip back up before we start making copies. The next enzyme we're going to talk about is topoisomerase, which is also called gyrase. Same enzyme, two names. The job of topoisomerase is to relax the supercoiling. You can imagine that if I am pulling apart this twisty thing, it's going to start to get lots of knots here where I have the untwisting trying to untwist really, really fast. So the job of topoisomerase here is to actually relax all that knotting that's going to happen as I try to unravel this twisty guy. Topoisomerase actually does this by literally cutting the DNA so that all the tension of all of that twisting can like unroll, and then it just zips that DNA back together. So topoisomerase, also called gyrase, is going to relax the supercoiling ahead of what we call the replication fork. Where I'm unzipping the DNA is the replication fork. Ahead of the fork, where I haven't copied yet, we have topoisomerase relaxing the supercoiling. The first weird thing that happens now is I have this enzyme called DNA primase, and DNA primase is going to come in and put down a little primer so that we can get replication started. Odd thing, DNA is a little bit high maintenance. It does not like to be at the start of the line. Its shape just doesn't let it be at the start of the line. And so we're going to start copying our DNA by making some RNA. Crazy sauce. So DNA primase is going to get DNA replication started by throwing some RNA in there. Those RNA nucleotides are complementary to the original DNA. It's not very large. We're looking at just 10 to 15 nucleotides, U's, C's, G's, and A's of RNA. We're just going to get that little chunk started so that DNA can come in and stick to the end of the RNA. Because DNA can totally be like 5th in line or 16th in line. It just doesn't be first because it has a weird shape. It doesn't like to be first. Once we have that primer in place, primase puts down that primer, then another enzyme, DNA polymerase 3, that's Roman numeral 3, is going to roll in and start attaching DNA, finally, to our primer. So here's polymerase 3, maybe our RNA is here, and then polymerase 3 is just going to start adding pieces of DNA to the end of the RNA. This happens in a 5' 5' to 3' direction. It happens on both strands of DNA, so kind of cool. We're going to copy both of the template original pieces of DNA at the same time. So here I have a strand and here I have a strand, and we're making copies on both of those guys. Both are happening in a 5' to 3' direction. But why? Why does it have to be a 5' to 3' direction? It's so cool. So here's our new nucleotide. Let's say that this guy is maybe guanine. And I need to throw this guanine in here because over here I've got my cytosine. So this original piece of guanine, before I add it to the DNA, is actually what we call DGTP. D for deoxy, G for guanine, T for triphosphate. Because before I add this guy to the new strand of DNA that we're making, it doesn't have just one phosphate group, it has 1, 2, 3 phosphate groups, kind of like ATP, but now it's GTP, but D for deoxy. Those 1, 2, 3 phosphate groups have lots of energy stored in them. And so what polymerase does is it chops off those phosphate groups, uses the energy from those phosphate groups to glue this phosphate end onto that 3' end of the growing strand of DNA. I need the energy from this phosphate in order to be able to make the bonds between the nucleotides. This is why we have to always copy our DNA in a 5' to 3' direction. I need to add the 5 of the new guy to the 3 of the old guy because 5 is where all the energy is stored. And so we're going to add the 5' end of the new nucleotide to the 3' end of the previous nucleotide, again, so that we can harvest that energy stored in the phosphate groups on carbon number 5 of the nucleotide. But what's crazy about going 5' to 3' on our leading strand, it's pretty easy for us to be like, here we go, and we're just going to keep going, and then helicase opens a little bit more, and helicase opens a little bit more, and it just keeps going. We call that the leading strand because it can be replicated continuously. We can just go and go and go. On the opposite side, though, because polymerase needs to go this way because of 5' to 3' direction, we end up making little chunks. We call those chunks Okazaki fragments. So we put our primer here. We're adding our nucleotides in this direction away from the replication fork because we have to go in a 5' to 3' direction. We have to go from 5' to 3'. We have to go that way. And so it's just a little bit of a struggle bus because then we open up a little bit more. We need a new primer and then more DNA. And then we have to open up a little bit more. And I'm going to have to put in a new primer and then more DNA. And so again, we end up with lots of fragments because we have to replicate away from the replication fork because 5' to 3'. We need the energy stored in the 5' end of the new nucleotide to be able to make those bonds between the previous nucleotide and that next nucleotide. Here's another image looking at that same idea. So our leading strands, those Okazaki fragments, those little pieces that pop up on what we call the lagging strand, all because of 5' to 3' direction. So here we have our helicase. Helicase is opening up that DNA. I put down that RNA primer with primase, and then polymerase 3 can just add DNA all along. Helicase opens up a little bit more. See, we have it opened up a little bit more. And polymerase can just keep going. We can keep replicating continuously toward that replication fork. Helicase opens up more, and then polymerase 3 can just keep going. It is leading strand. It is copied continuously. It can happen a little bit faster because we don't have so many pieces and parts. Because on the lagging strand, so many pieces and parts. Those fragments are called Okazaki fragments. So what happens is helicase opens things up. We're going to put down that little bit of primer, and then we have to add DNA moving away from that replication fork. Helicase opens up a little bit more. Another primer has to start here. and then we're going to add the DNA with polymerase heading away from the replication fork. Polymerase is going to add DNA until it runs into the RNA primer that we had up here, and the whole thing has to happen all over again. Helicase will open up a little bit more, we'll throw that primer in, and then polymerase will add DNA that way until it runs into the primer that came before it. And then she's going and going and going. Leading strand can be copied continuously toward the replication fork, 5' to 3' direction. That lagging strand is copying the DNA away from the fork in these tiny little pieces called Okazaki fragments. Now you might be wondering if all of our DNA has a bunch of little pieces of RNA in it because of those primers. And the answer is actually no, because we have these other super cool enzymes that come in and take out the RNA primers. And then this guy, DNA polymerase 1, is going to fill in those little gaps where the RNA used to be with some DNA. So exonuclease and DNA polymerase 1, these guys work together to pull out the RNA and plug in that hole with some DNA. But polymerase 1 cannot glue together those funny little pieces where the primers started and stopped. And so we have one more enzyme come in, DNA ligase. Ligase's job is to bind together those DNA fragments. So we have these tiny little holes left over from where the different primers used to be, where we had to take them out and then fill in those gaps. Ligase is going to come in and glue those guys together with one last little bond so that we have some continuous pieces of DNA. And that is DNA replication. We humans have stolen DNA replication to use in technology, and we call this PCR, polymerase chain reaction. Polymerase, because that's the name of the enzyme that copies the DNA. Chain reaction, because we keep doing it over and over and over again. It comes in 3 stages. The first stage is denaturation. Remember this from proteins. If proteins get too hot, they lose their shape. Super cool thing about DNA double helices, if we heat up the DNA, these guys wiggle and jiggle a little bit too much, and so the hydrogen bonds are not strong enough to hold together super wiggly DNA. And so we end up with denatured or melted DNA. It's not really melted, we just took apart the double strands. So now I have some single-stranded DNA. We're going to anneal or glue to those single-stranded pieces of DNA some primers. So we pick what gene in the DNA we want to copy, and we create a primer, a little piece of DNA that's going to match. We anneal these primers to our open strands of DNA, our single-stranded DNA, anneal those primers, and then we elongate. That polymerase enzyme comes in, attaches to the ends of the primers, and then makes DNA complementary to our original strand. So at the end of our elongation stage, we have 2 pieces of DNA that are matches for that original piece of DNA. Of course, in order to make this happen, I have to have lots of A's and T's and C's and G's floating around in my reaction tube so that we can actually make those copies. What is crazy about this, my friends? The whole process, steps 1, 2, 3, only take about 2 minutes. So in 2 minutes, I can go from 1 piece of DNA to 2. 2 minutes more, I can go from 2 to 4. Every 2 minutes, we double the amount of DNA. So if I run this for 30 minutes, I'm going to have 15 to the power of 2. I don't have my calculator nearby, but that's a really big number. So we can make crazy amounts of DNA, big copies of the DNA, so that we can run forensic tests, or we can run hereditary tests, or we can run genetic tests. And this will allow us to have lots and lots of DNA to run those tests, because we wouldn't want to run out of DNA in the middle of a test. The enzyme that we use, the polymerase that we use in polymerase chain reaction, is called Taq polymerase. Taq stands for Thermus aquaticus. There's the T, there's the AC. So Taq, or Thermus aquaticus, Polymerase is a polymerase enzyme, DNA replication enzyme, that we humans stole from, you guessed it, a bacteria that lives in hot water. The reason that we choose Taq polymerase is because that's really hot. Remember that first stage? We had to melt the DNA. We're talking about almost boiling temperatures. If we were using human polymerase, we love a 37 degrees Celsius. Our polymerase would be denatured. Not just the DNA, but also the polymerase. But this guy, Thermus aquaticus, he loves hot water, and so his polymerase works the best in hot water. And so we can heat up that DNA, melt it, polymerase will not melt, and then we can make all the copies of the DNA that we want. And again, this is just showing this lovely, in just 4 cycles, so about 8 minutes, we can have this DNA, and now we have this DNA. Crazy cool, right? Amazing, amazing. And on that exciting note, we have accomplished our objectives. We reviewed DNA structure, double helix, complementary base pairs, anti-parallel structure. We talked about Meselson and Stahl. They used some nitrogen 15 and nitrogen 14 to figure out that DNA replication is semi-conservative. We looked at some of those enzymes involved in replication. We talked about helicase. We talked about topoisomerase, also called gyrase. We talked about DNA primase that actually throws some RNA into our mix. And then we have our polymerases. We have DNA polymerase 3 and 1. We have that exonuclease that helps polymerase 1 take out the primers and throw in some DNA. We wrapped up with that little look at ligase, which glues together all those little fragments. And we also talked about the amazing piece of technology, PCR, polymerase chain reaction, where we can make lots and lots of copies of DNA super, super fast. Good work today.